So as Dr. Bethke pointed out, I'm an aquatic toxicologist. I study the effects of both contaminants and climate change, and also the interaction between contamination and climate change on the effects of aquatic biota. I mostly focus on fish, and particularly within that on the early life stages of fish, because they are the most sensitive. And then one thing I wanted to point out before I get started that I think I forget to mention at times is that the approach that my lab uses to doing toxicology studies is that we always use environmentally relevant concentrations. So that means we're not doing count 'em and kill 'em toxicology. We actually use just the really low level chemical exposures that any fish would experience if it was in a lake or a river or in an estuary. So everything I'm about to talk about today was done in that vein, and it's all the exposures we've done reflect current environmental concentrations. So now I'm going to try to advance this slide. I have to click back on the thing. Got it. Yes. Okay. All right. So first of all, what the heck is metformin? Well, it is the first line therapeutic for type 2 diabetes, also polycystic ovarian syndrome, and then a few cancers where insulin resistance is a factor. And what I mean by first line therapeutic is that a doctor must prescribe metformin before they're allowed to try any other treatments if you want insurance to pay for it. So I'm sure you can imagine that means there are a lot of people on metformin. So again, within what my lab studies, one of the things we study is metformin's effects on fish development. And so the driving question, the focus of today's talk will be, do current concentrations of the diabetes medication metformin pose a risk to wild fish populations? And so you'll notice that I've underlined this word risk, and I want to clarify really quickly why I underline that and what exactly I mean there. So just take a moment and look at this slide and pick out the two things that you think would be the highest risk and the lowest risk. Okay, everyone have an idea preliminarily? Okay. So this is a graph of all of the numbers of deaths caused by each of these things. They're enveloped within here. The lowest risk things are terrorism and natural disasters. They're at the very bottom. So if you picked those, there's a good reason why, and I'll explain in a moment. The highest risk is air pollution and diet and lifestyle. And all of these things in green correspond with deaths associated with a sedentary lifestyle and poor nutrition. And all the things in orange correspond with exposure, chronic exposure to air pollution. So if this is you, and you're like, oh, that's because you were thinking about hazard. So hazard is different than risk. It's a component of risk, but it's not exactly the same. So hazard is something that is inherently dangerous. All of those things were hazardous on that first slide that I showed you, and you were probably ranking them according to their hazard level. But that's not the only component of risk. And so once again, we're going to assess risk of diabetes medications to fish, right? So this is a really good example, I think, that kind of does a good job of delineating the difference between hazard and risk. So on the left here, I have a table that shows the projected casualties and incapacitations that would happen in a city of 500,000 people if there was a bioterrorism attack with anthrax. Okay, so the researchers project that about 20% of people will die immediately during that attack, and about 25% will become incapacitated. So that's about 45% of the population, right? On the right, we have the CDC flu burden estimates over a 10-year period of time. About 0.02% of people died in the United States during that time. 0.23% were hospitalized from the flu, and 13% got the flu and were ill. So looking at these two things, you can see that one of these is much more hazardous than the other, and which is the most hazardous? So is bioterrorism hazardous? Absolutely. Is the flu hazardous? Can you die from the flu or become seriously ill? Absolutely. Show of hands, who has had the flu? Who has died of a bioterrorism attack? None of you. And there are actually no recorded incidents in the United States of anyone ever dying from a bioterrorism attack. So there have been attempts, very small ones, infrequently. No one has ever actually died from anthrax. And so that is the difference between exposure and risk. And that is, I'm sorry, between hazard and risk, and that factor is exposure. So risk is a product of both how dangerous something is and how often you're exposed to it, or how much you're exposed to. Those two things must be present. And so the takeaway here is, if there's no exposure, even the most hazardous thing cannot pose a risk to you. But as exposure increases, things that would be seemingly benign become, in fact, very risky to you, okay? And so that's what I want you to keep in mind as I proceed through these things. So with this background knowledge, let's go back to our driving question about whether metformin poses a risk to fish. And to answer this, we now know there has to be a hazard. So metformin must adversely affect the health of fish. And then it must be present in high enough concentrations to cause that effect in fish in the environment. So we're going to examine whether those two conditions are met and see if we can answer this driving question. Exposure is the easiest one to deal with first, so let's start there. This is a map showing global diabetes, type 2 diabetes trends. Keep in mind that you have to try metformin before anything else if you are a type 2 diabetic and you go to the doctor. That is the first thing they're going to give you, right? So globally, rates are skyrocketing, although they're highest in countries with really high incomes that are very economically developed. That includes us. And so if you're like me, you automatically want to see a map of where you live. And so here it is. About more than 11% of Americans have type 2 diabetes, which is a massive, massive proportion of our population. And then if you look at the Texas coastal bend here, we are not doing so hot in terms of the prevalence rates. They're very, very high. So another factor here is not just how many people are taking the metformin, but as Dr. Besky pointed out, how much of that is being excreted from the body and making its way into wastewater treatment plants, right? So metformin is funny in that 100% is excreted unchanged in your urine. So almost the entire dose is excreted unchanged. It's not metabolized by your body. It causes a biological effect, but it leaves your body unchanged. So that means that every dose that is taken by a person ends up in the toilet at some point throughout the day, and that goes into wastewater streams. So once it gets into wastewater streams, microbes can metabolize it. And they can metabolize it into a bunch of weird stuff, but the only one we actually care about is called guanylurea. And the reason we care about that one is because it sticks around. The other ones are pretty fleeting. They'll be broken down into something else, but guanylurea hangs around for a long time. So because there's so many people on this drug and because it's excreted unchanged and the whole dose is excreted unchanged, it is actually the highest drug by weight of all drugs that is released to the aquatic environment. And when you have something that is being put into wastewater treatment plants in such large amounts, it doesn't matter if the metformin gets broken down to guanylurea quickly or not. It's always being renewed, right? There's a constant supply coming out of a wastewater treatment plant, which we call pseudo persistence. And what that means for fish is that they can expect to be exposed throughout their entire life cycle. This is a normal occurrence in any receiving waters that are downstream from any kind of wastewater treatment plant, including our estuaries. And so surface water levels, if we look globally, are for metformin, they're as high as 34 micrograms per liter. That probably doesn't mean a whole lot right now, but it will later. And micrograms per liter is parts per billion. And so guanylurea has actually been measured up to 222 parts per billion in surface water. So this is not the effluent coming right out of the wastewater treatment plant. This is in a river or a lake or an estuary, where it tends to be lower, but so it's probably a freshwater environment. But the point I'm making here is that these are actually pretty high concentrations for a drug. And I'll explain why that is important in a second. But also what I want you to look at is the black represents guanylurea relative to metformin. So you can see that a massive proportion is actually present in that guanylurea form, not in metformin. And that was the one that the bacteria transform into, which becomes important later in the talk. So I said a lot for a drug. Why does that matter? Well, it matters for several reasons. Why are drugs designed? They're designed to cause a biological effect. They're extremely potent. It's not as if it's your conditioner or some other kind of chemical that's being washed down the drain. It was just gonna, be a fleeting thing used for a different purpose. These are actually engineered to cause a change in your body at a very low dose. Now, it's a human drug, so why would it affect a fish? Fish are vertebrates. And a lot of the biological pathways that get affected by common drugs in humans are also present in every other vertebrate. And so a lot of the drugs that we take do the exact same thing in animals, particularly fish. So another thing that we want to keep in mind is that guanylurea, the transformation product of drugs, are more often than not also metabolically active, meaning they cause the same or greater effect in people or in fish than the parent compound does. So all of that guanylurea can do the same thing as metformin. And then this is another thing that we don't tend to think about is how many drugs are in wastewater treatment plant effluent and constantly going out into the environment. And a lot of them have similar mechanisms of action, meaning they act on the same pathways in people. And so the fish are exposed to this complex cocktail of all these drugs at fairly low levels, but together, we don't really know what it's doing to the development of fish. It's really kind of a giant unknown and a really difficult question to answer. So from what I've said, we know some things so far. We know that exposure is probably going to happen, right? That's there. So we had those two components of risk that included exposure and hazard. And based on the data that we find, we know that exposure, we can check that box, right? And then I want you to keep in mind that early life stages, including embryos and then larval fish, are much more sensitive to the effects of chemical exposures than other life stages. So we know there's exposure, now we have to determine the hazard. And so that can be done through a couple ways. That's what toxicity studies aim to do, is determine the hazard of a compound. And so you can do that in a lab study, or you can do it in a field study, or you can do it in a combination of both, which is what we're going to talk about when I present my data later on. But what I'm showing here is, I'm showing it for a reason. So these are racks and racks and racks of zebrafish. And these zebrafish colonies are maintained in labs under perfect conditions for generations and generations and generations. Why would you choose to keep a particular fish in a lab? Would you like to, why do people, let me ask you this question. Why do people keep freshwater tanks rather than saltwater tanks more frequently? They're easier, right? So we have a really, really easy to care for fish in perfect conditions in a lab with no predators. They're fed every single day. There's no food shortages. There's no temperature variation, just ideal conditions. It's like staying at the Hilton for these fish. And these fish are already pretty tough because we chose them specifically because they weren't going to die on us all the time. They're easier for the grad students to take care of, right? And so that's important to keep in mind because this is the type of study that EPA uses to develop the water quality criteria that protect aquatic life, is the values generated by these standardized lab studies. And we'll see why that's important when I present the data later from my study. The other one is a field study. This is much more realistic, but how do you know it's the metformin? I just said there's like a cocktail of things in the water, right? And that was just pharmaceuticals. That doesn't include all the industrial chemicals that are going out into the water. How do you decide what's causing the effect on fish health when there's just so many things there? It's difficult to make an association, right? So what do we know so far about hazard in metformin? Well, from lab studies, we know for the most part that microgram per liter, single microgram per liter concentrations of metformin can affect the growth and of early life stage fish. And that includes a couple of different freshwater species, including Medaka and fathead minnow. We also know that it affects hatch success of those fish, and it affects the behavior of the early life stage fish. Interestingly, in adults, it also causes feminization. So what I mean by that is that the males in their gonad tissue actually start to produce oocytes or eggs. So we actually see physiological changes in these fish that correspond with masculine fish transitioning to a more feminine phenotype in several different ways. And they'll also lose their secondary sex characteristics. And in this particular study, that would include these fish get really dark bands when they want to mate and guard their nest, and they get all manly and tough and aggressive acting, and they stop doing that when they're exposed to environmentally relevant concentrations of metformin. There's also a bunch of molecular data that supports all of these changes that we see. We don't really need to go too deep into it, but I do want you to know that these aren't just things that a few people observe. There's been a lot of probing into the mechanisms behind these. Now for guanylurea, we see the exact same thing. The difference is this right here. These are the changes. So anything with an asterisk is a statistically significant change relative to the unexposed fish. Okay, so this is showing growth in early life stage fish. So anything exposed to 1 part per billion or more of guanylurea had stunted growth. Now, if you look at, metformin, if you look at guanylurea, the units change. Anything exposed to 1 nanogram per liter or more, which is a part per trillion, which would be a single drop in an Olympic-sized swimming pool, that will cause a change in the growth of these fish. And so the take home here is, I mentioned earlier, a lot of times the transformation products of these pharmaceuticals are metabolically active, and that is definitely the case here. And in fact, it's not just active, it's much more active. It's very, very potent. Again, we have a lot of molecular probing tools, looked at this. Data is all supported. This is actually a really interesting graph, even if you're not a molecular person. This is showing essentially the feminization effects that we see in the proteome of these fish. So the males that were exposed to guanylurea had similar proteomes, or they had similar protein production in their bodies as the unexposed females. So that confirms that feminization starts happening during development, not just later in life. So again, these conditions aren't very realistic, right? I mentioned this is a lab study. So is there really an ecological risk? Because we know there's some indication that hazard might be present, but we're not sure because we haven't tested this in an environmentally realistic scenario. And so this paper here by Caldwell et al. Decided to use modeling approaches to try and figure out if in the environment, fish would even be able to be exposed to these compounds, because they hypothesized that it would partition to the sediment and that it would become sort of buried and the fish wouldn't come in contact with it. So yeah, it's there and maybe it would cause an effect in the lab, but it's probably not going to in the field. So we have this disagreement here between these two approaches, between the lab studies and the modeling studies. And so again, we can't really answer that question about ecological risk or not, which involves a whole bunch of math. But if you look at the predicted safe concentrations from that Caldwell et al. Modeling paper, They basically showed there's absolutely no possible way that enough guanylurea or metformin can be released to the water to ever cause a problem. That was their main conclusion of the paper. So I keep mentioning that in the field, conditions are different, right? So what am I really talking about? Well, there's species sensitivity differences. There's life stage sensitivity differences. So again, not all life stages are equally as sensitive to chemical exposures. Not all species are. And this one's really important. Those zebrafish kept in those racks at the Hilton in the lab, they don't have the same type of sensitivity as the zebrafish, which aren't really a very environmentally relevant fish to begin with. They're only like in the Ganges. And then also there's like a population in Florida that was someone's pet that got released and survived somehow, but I digress. Anyway, so the wild fish are going to face a whole bunch of different circumstances, and their sensitivity is also probably going to be different, because if you think about dogs, they all originated from wolves, right? But we have selectively bred them to have different characteristics. We've done the same thing with our lab fish. We have selectively bred them to be tougher and bigger and produce more eggs, because it's easier for us to do studies if we have more eggs at a time, right? So our fish that we have in the lab might be the same species as the fish in the pond, but they're not the same. They're essentially different breeds. And then you also have effects that can happen lower down in a food web. So there's a really famous study done at the experimental lakes area. I don't know if you guys might remember. Acid rain was becoming a really huge problem, wiping out forests. They were doing all these lab studies, and they were acidifying the tanks that the fish were swimming in, and nothing was happening to them. They were fine. And they were like, acid rain's not a problem for fish. But then they did a whole lake study, which is where, actually, I'll show you a picture of that later. It's where I did this study. They acidified an entire lake in Ontario. And what happened was the base of the food web collapsed and the fish starved. And that's why acid rain is very, very harmful to aquatic ecosystems. It's not because it directly impacts the fish, but it kills all their food and they starve to death. Okay, so I mentioned we are going to present data, so I'll get to that. We basically wanted to do a lab study and a field study to compare and see whether the values that have been generated that should be safe for fish in that Caldwell et al. Modeling paper, and then also just in laboratory studies, we wanted to see if those were real in a wild and realistic setting. Now, I mentioned there's a whole bunch of problems with lakes. If you just go out and catch some fish, how do you know what they've been exposed to? So we had a solution. And those solution is whole in-lake mesocosms. So essentially, this is at the experimental lakes area in Ontario where they did the acid rain study. You have these floating collars, and then you have an impermeable curtain that you suspend and you seal it to the sediment, and then you spike it with whatever chemical it is you want to expose the fish to the exact concentration you're interested in looking at. And you load it with a known number of fish. And by that route, we can then kind of do a field study, but keep all of the food web present in the water column. We can keep all of the natural organic matter. We can have interactions with the sediments. So if, in fact, as Caldwell et al. Predicted, it was all going to go into the sediment and then the fish would be safe, we would see no effects, right? And so that was our solution. I'll be referring to mesocosms throughout, and that's these floating tanks. Essentially, they're about 10 meters across. They're very large. And then we also did it in the lab. So all of this was done using the same species, once again, a fish, fathead minnows. And we essentially just looked at all of the endpoints that we could on the early life stage fish health. So I mentioned the cool acid rain study. This is another cool study that was done in the experimental lakes area. This was when we found out that it was phosphorus was the limiting factor in algal blooms. So they literally divided the lake in half, and they didn't know what about fertilizer was causing lakes to become full of algae. They dumped nitrogen in half and phosphorus in the other, and they waited to see. And this is what we saw. So this is a really, really famous study that was done that essentially resulted in a lot of policy changes thereafter. So this just shows all of our replication and stuff. There was a lot. I'll leave it at that. Oh, my goodness. We also did a lot of stuff with the adult fish, but I'm just going to focus on the early life stage fish results. Again, I kind of already explained this, but we did the field setting with the wild eggs from the wild fish populations, and then we used our lab populations. And an important component here that was different was that we used water that had been part of the lake ecosystem, right? So we spiked it. There are microbes in there. They can transform it. It could partition into the sediment, or it could stay in the water column. We just didn't know what would happen. So we spiked that water. We left it in there for several weeks beforehand, and then we did exposures with it. So in this scenario, we just used our special fish from the lab and our super clean water from the Hilton. And this is what we looked at. We looked at hatch and survival. We looked at size from a couple of different endpoints. We looked at their eye formation. We looked at their heart and yolk sac edema, whether their face and their jaw formed normally. So all these things really impact whether a fish can survive in the wild or not. If you have a malformed jaw, you're not eating, right? That's a big problem. We looked at spinal deformities. You can't swim if you have a deformed spine. And so all of these things were measured in each of the fish. This is obviously a rather unhealthy fish that was exposed to metformin. This is yolk sac edema here. And this is where this poor little fish's heart should be. So that's the heart of this one. And this is the elongated tube heart on this fish, which means it's not going to be able to deliver oxygen. So it's alive for the time being, but it won't be for long. And so we refer to this as ecological death. So these fish are technically alive, but they're not really going to ever have a chance of contributing to the breeding population. So what were the differences we saw? A lot. Just to begin with, the minnows, the fathead minnows that we had in the lab, remember I said that it was kind of like selective breeding. They were over 3 times larger. Just the control fish, comparing them, they were over 3 times bigger than the fish from the wild caught population. So automatically right out of the gate, there's some pretty important differences there. We also found that their surface area was 10 times larger. Their eye size was 10 times larger. They had about 10 times more surface area on their yolk sac, which translates to energy for fish to develop. So really, we expected from the beginning that, in fact, the effects were not going to be the same on both populations. And so just showing here, there weren't actually effects on growth in the wild population, which was a surprise to us. There were effects on the lab spawned population. They got bigger. So I don't know if you guys know this already, but the size of an early life stage fish is the primary determinant of whether it lives or dies. So the bigger you are, the better your chances of surviving to breed. And so these fish actually, from a biological fitness standpoint, did better if they were exposed to metformin than their counterparts that were not, which is very strange. So again, not what we expected to see, but they were longer, but they were skinny. So I don't know, it's kind of a trade-off. Who knows? We don't know the fitness implications here, but we know there is a change. And so that still remains to be seen. Either way, the wild fish were not affected in size. Eye size is, it was much smaller in both of the population, in all of the metformin exposed fish in both populations. Why does eye size matter? Well, first of all, if you can't see, you can't eat. That's one thing. But it's much more indicative of a larger problem to do with energy consumption. So does anyone know about the Mexican cavefish that don't have any eyes? Yeah, maybe. So there's a Mexican cavefish that doesn't have any eyes. And there's a reason for that. Fish in really nutrient limited situations, one of the first things that they start to get rid of is their brain tissue and their eye mass. And there's a reason for that. Those are the 2 most energetically costly things in development. So if size is the most important determinant of whether you live or die, who cares if you're really smart is essentially sort of the premise here. It's a trade-off, right? Like what's most important to get you through development and into a stage of life where you have a better chance of living. And it's a growth lengthwise, not with your eyes. So we see that metformin does lead to much smaller eyes, significantly smaller eyes in all of these. Oh, here's my Mexican cave fish from a cave, same species, not from a cave. They got rid of their eyes entirely because there just isn't enough nutrients in these caves to sustain neural development. So I already explained all of this stuff, and we're moving. Oh, well, this is actually, I'm going to come back to it. So yolk sac size was significantly smaller in the metformin exposed fish that were from the wild population only. So there was no change in the lab spawned population. And we see that this sort of corresponds with that eye shrinking situation. So they're having trouble completing their energetic and metabolic processes that drive development in the wild population only. The lab fish seem to still be fine. Even though they had smaller eyes, they didn't really have any odd changes to their yolk sacs, which is their energy stores. So that was very different between the two. And then this is kind of a really busy figure, and you don't need to look at all of it, but what it's showing you, really you just need to look at the colors, because the colors indicate whether something was it positively correlated or negatively correlated. And if you look at this is the wild fish. There are way more dots here, right? Meaning way more things were impacted by metformin exposure, and they were not impacted in the same way as in the lab spawned fish. So effectively what this is showing us is that these lab spawned fish that we're using to develop all of these regulatory criteria to protect fish are not accurate. They're not doing a good job of predicting actual risk for wild populations of fish, which is pretty problematic, I would say. We talked about this being really important in terms of energy use in these early life stage fish. And if you think about the effects on the small yolk sac and then the small eyes, if you don't have enough yolk sac stores to complete development, what's your next option for getting energy? It's not a trick question. You have to go hunt, right? But a lot of these fish could not see very well, and they also had spinal deformities. They had craniofacial abnormalities, weird jaws. So there was no way that they could actually eat. And they had to eat because they were running out of yolk really fast. So if you think about the larger fitness implications here, what's really important to take home is that just because something successfully hatched doesn't mean it's alive. It comes back to that idea of ecological death, where once it goes out into the wild, is it really part of the gene pool? And the answer here is no, in a lot of cases. There were a lot of very severely deformed fish. So, and once again, we saw absolutely no deformities in the lab spawned fish relative to the controls. It was only present in the wild spawned fish. And so as I start talking about the deformities that we saw, I'm only going to be talking about the wild population. This is kind of the same idea as before, just showing those different patterns and different directions of trends. I showed you the picture in the beginning of the edema in the fish. And so this is showing just a figure describing what you visually saw in the first slide when I showed you the difference between the heart size. And then over here, this is looking at the increase in deformities in the eyes, the jaw, the head, the yolk sac, and the pericardium, all broken down. And there is a dose-dependent response. So as the metformin exposure increases, so too do the number of deformities in the wild population. And our lab fish just stayed happy and kept living their best life. And so again, this is another way of showing the severity of deformities. Another thing to point out is that these fish had co-occurring severe deformities. So it wasn't just their jaw was deformed. It was their jaw, their eye, and their spine were deformed. In most cases, fish had 2 or more severe deformities. Again, showing the change in the direction. I'm talking over time, so I'm trying to go really fast. So in conclusion, essentially, we see that there is a very big difference in the effects of these chemicals on our different populations of fish. So it makes a big difference, the conditions under which fish are exposed to these chemicals, whether or not you can get toxicity values that will actually protect the fish once you put them into regulation. And then finally, we did observe that there was guanylurea formation in the water in those tanks out in the wild. And so that's a really plausible explanation for why we saw more severe deformities in those fish, is because they're being exposed to guanylurea as well as metformin. And if you'll recall from the very beginning, guanylurea was a much more potent chemical relative to metformin when we exposed those fish in the lab. So that's a potential explanation for why these wild fish had such greater responses than the lab fish. And so I think I pretty much just summed everything up, and I'm going to take questions really fast and thank all my funding agencies and collaborators. There were a lot. Thank you. Yes.
How do you anticipate these findings impacting the FDA's approval process?
I don't, but what I do anticipate is that if you can show serious risk of these chemicals, the wastewater treatment plant process will be required to change, because what gets regulated is not, the regulations don't change to protect fish on the FDA's part. That's just to protect humans and see if the drug is going to do what it says and not give, you know, undesirable side effects to the humans. But the EPA is the one who says your effluent can't contain more than this, this, and this, or it will harm the ecosystem. So I don't expect that it would, the FDA will care all that much, what I have to say. But hopefully, with studies like this, the EPA will start to see that just using these lab studies with these ideal conditions for fish is not going to give you a really good idea of the actual risk, and they can hopefully sort of adjust some of these regulatory criteria that they've created. Anybody else?